We'll then move on to the next speaker, Lin. Dr. Lin got his PhD in Iowa State University in 1997 in the field of animal breeding and quantitative genetics. Right now he is an associate professor with the National Taiwan University in similar fields: bioinformatics, genomics, quantitative genetics, population genetics, and animal breeding. He's going to give a presentation to us on SNPs and protein markers for reproductive traits, and in particular early embryonic developments in the trace kids. So Dr. Lin, floor is yours.
Good. It's my great honor to be invited as a speaker in this conference. Especially, I have a chance to talk to Dr. Nagai about our development of research, especially in sow reproductive performance in Taiwan. Because in Taiwan we have a little bit different production system. Before we have before we said 3 breeds crossbreeding system, but now we change a little bit from 3 breeds to 2 breeds, but combination, like Landrace with Yorkshire as the female, that's 3 breeds, and later on the terminal sire is Europe. But now after the 1997 outbreak of FMD, we changed from back to the domestic market. So we use the Landrace as the sow more than Yorkshire. So we have another system like a 2 breeds crossbreeding system. Landrace sow directly mated to the terminal sire Europe. So that's why we have this kind of research, especially working on the Landrace. Not only doing the selection program, like traditional, we can call it traditional, because it's been applied for many years. Select on the reproductive performance of sows, then select those best performance sows, then make it to boars to have great or potential reproductive performance yields. And we applied another way using functional genomics in the beginning. Actually, I'm going to talk about a series of studies been around 10 years of research, from the functional genomics study and to single nucleotide polymorphism, SNP markers for selection later on. And also we developed protein markers for checking the female reproduction performance on farm directly. We know the reproductive success is always an important indicator for pig production. And one of the important major components of reproductive performance success is litter size, especially for the farmers. They need to have a larger litter size to have more pigs for production efficiency. Unfortunately, litter size is a very lowly heritable trait. It's just 0.1, 10% or 15% heritability. So it's not easy to select to get the progress of swine, the genetic progress. Also, it's a sex-limited trait. So it's difficult to select the boars because they don't have any female reproductive records. So it's we try to do it another way. We know that genetically what kind of genes is expressed during the embryo, early embryo development. Then we start working on those genes, the variation of the genes, and then applied it to the farm, the pigs on farm, to see any kind of improvement we can find by this way. We know 4 major developmental events during zygote to blastocyst. After blastocyst, we know that the embryo moved to the uterus and locate there and as a more mature embryo. So during that early stage, before implantation in the uterus, the first event is maternal to zygotic transition of the transcriptome and the proteome. And the second one is to the medulla. We know that the cells, the embryonic cells, compact together and move from that part by cell-cell interaction and genetic determination. The third one is the first tissue differentiation of the medulla to blastocyst. So the last one is the maternal embryo interaction in the blastocyst development. Like the baby informs its mother to prepare the uterus for them to locate. We started actually before 2005. It's a long story, so I made it short. So I made this flow chart and make it easier for everybody to understand. We actually start the whole thing, not only the pigs side, we also have the chicken side. 2 different teams, but we work together because the sequencing, the cloning, these things, we work in the same labs or the similar groups. We can do it more efficiently. And doing the sequencing, then we have the same team for bioinformatics to annotate which genes expressed actually. And later on, we have microarray expression profile experiments, and the monoclonal antibody production, and the microRNA prediction, that kind of things. For the chicken side, we have microarray also, and SNP prediction and validation. For the landrace side, we know we totally find out, actually not as this much, more than that. Actually, we found about 6,000, but we annotated with function around 3,000 genes expressed during that time, from 4-cell, 8-cell, medulla to blastocyst. And we annotate what kind of functions they have. We come out 4 categories of functions for this around 1,500 genes. Cell activities and the metabolic pathway, that's the major parts of the genes function. And then immune function is the least part. And the regulatory mechanism, total about 1,300 genes being annotated at that time. Right now, we already increased the numbers to more than 3,000 already. That's some of the genes I listed here. Actually, we already have a lot, but because too many, so we need to find out which one is the key person. Then we developed a homemade 24K, like 24,000 genes, spotted on a microarray, a small piece of slide, especially designed for porcine embryo development. And we got not only from the genes, not only from our research, also from the NCBI unit genes, those being found in embryo, those genes being found. And we compare, one group of us compare medulla embryo and the blastocyst embryo. We collect from different high reproductive cells at this age. And the majority we found significant differential expressed results related to gene function of metabolic pathway and cell activity. We actually found those differentially expressed genes actually fall into these two groups. That's one of our publications on the Journal of Reproductive Domestic Animal, Reproductive Domestic Animals. And we have the manula, the manula expressed more than blastocyst, and this part is the blastocyst expressed more than manula. And we found that the percentage of the genes expressed differentially is more in about 70% genes display differential expression is blastocyst more than manula, and the manula more than blastocyst only 2.36%. And the others unqualified or equal. And for this part, especially much higher expression in manula is 162 genes. And for blastocyst, more expressed more in blastocyst, 5-fold change than in manula, then it's 2,126 genes. Okay, that's just a short list of these 162, just a few of them. And later on, we start another research about single nucleotide polymorphism markers, searching on that. And this part is for the blastocyst side. And we also checked using the qPCR to validate these 12 genes actually really express the microarray data. Okay. So we make sure it really express more in for this more in manula, and this part more in blastocyst. And that's the paper we already published in 2012. I think, yeah, 2012 in reproduction in domestic animals. Okay. Then we started the continuous research on single nucleotide polymorphism, the SNP markers, looking for the new markers for selection, because we know that that's a lowly heritable trait, and we might not just depend on one marker to select, like most of the marker-assisted selection. We need to combine a lot of markers put together to select on. Maybe we can gain more progress on cell reproduction. And so we need to identify even not only the QTL, the candidate gene approach, or we so-called marker-assisted selection. We need to identify the new polymorphism in different genes. Especially those genes should be on the same pathway or same pathways. Yeah, several pathways probably together affect the cell reproduction, because the cell reproduction procedure is a long procedure from zygote, 1 cell, 2 cell, 4 cell, 8 cell, to manula to blastocyst. Also, after that, after implantation, they need to develop to be a fetal, and then come out as a piglet. So it's a long 114 days development. So there must be a lot of genes involved in this process. Oh, sorry. And those SNPs in our study, we focus on promoter region, because this region concerned about the transcription, the expression efficiency. And before that, we have the microarray differential expression profile. So we think probably this area may have those genes we found in the previous study, the microarray study, we tried to find out the single nucleotide polymorphism on the promoter region, because this region concerning about the efficiency of expression. So we focus on this at first. There's the more than 20 genes in this list, not only from our study, and also we have metabolic study, metabolism, the lipid metabolism in embryo study for a long time. So we know some of the genes being critical, playing critical roles in the metabolism in the embryo. So we try to include them, some of them here. Yeah, there's the adiponectin, the vastatin, and also the perilipin and the serum amyloid A. And also from the literature, we already know the prolactin receptor. That's one of the genes being found effective, affecting the cell reproductive. And also prolactin, leptin, and we call it RBP4. There's the retinol binding protein 4. That's from the literature. Also, we found some variation in the promoter region as well. Actually, we have another one. I didn't put it here. The estrogen receptor being found many years, like 1980 something. Yeah, when I was in Iowa State University, yeah, it's been found. And but the gene, the polymorphism being found effective in Yorkshire breed or Chinese breed. So we found that ESR are not effective here in Landrace. Also, the polymorphism not easy to find in Landrace population. So we just actually we included, but we found no effect on our study. Okay. When we discovered the SNP on the promoter regions of these genes in Landrace cells, we found 30 Landrace cells from 4 different purebred herds in Taiwan. And we select 15 cells from these 4 purebred herds with high total number bone trait. In average, these 15 cells with total number bone is 14.5 picometers. And another group is the same 15 cells from the same 4 different purebreds. In average, they only have 7.4 picometers. We found the SNP in those genes promoter region, such as this. For each gene, we found several places or just one. Yeah. And the genetic evaluation, we come out to the more Landrace cells and with records, totally 456 Landrace cells with 2,443 farrowing records from 3 major purebred herds in Taiwan. And we apply mixed linear model like a graph, this kind of to estimate the breeding values of the total number bone and the number bone alive and the average birth weight for the cells. And the 2 groups of finally we have 2 groups of 90 cells, each 90. So highest 20% EBV cells. We have 90 cells for, we call it top 20%, 90 cells in this group. And another 90 cells in the lowest 20% EBV group. We call it a bottom 20% group. And we apply another statistical method like a test control association test. It's a chi-square test. This kind of association study method in statistics being applied frequently in human disease, like normal as a control and disease affected as a case. Or in cancer development, they have like a more serious development and normal or in between. That kind of study, we call it case control associations test. So I don't get into the detail of this formula. And the results, just part of the results here we list. The T20% in total number born, this trait is the EBV, average EBV is 1.29 plus minus 0.33. And the bottom 20% is minus 0.88 plus minus 0.34. What does it mean of this kind of EBV? 0 is the average. So that means this group is 1.29 picolis more than the average. Okay. And the bottom 20, the minus 0.88 means less than the average with 0.88 picolis. So we try to do this as the case, this as the control. So we have the chi-square test with the significant one. We list 4 of them here. That's the ATM gene, two different SNP. And here is the F-box 032, one SNP on it. And that's another gene, we call it oxidative nitro domain containing protein. Working on the cell cycle, apoptotic process, and the transcription also. Here I put the red letter here. It means that favor the TNB. This T type, T allele, favor the TNB. And also this G allele in this ATM K SNP marker site. We call it G allele is favor top 20. It means favor the total number born, and the other the same. Totally, we found 17 SNPs at the time on promoter regions of 8 different genes with significant difference between the two groups. And right now we're getting more results like this. And that's for the number born alive. I think the same scenario here. ATM still play important role, and another two I list here. SAA, that's the serum amyloid A, is a positive regulation of cell adhesion, protein secretion, and the immune response. And the LASP1 is LIM and SH3 protein 1. That's positively regulate the signal transduction, cell proliferation, that kind of things. Okay. So totally 20 SNP, new novel SNP being found for in 8 different genes affected number born alive. And the average birth weight, we found the same scenario, like we totally found 14 SNP sites on the promoter region of 8 different genes with significant difference between the two groups. And here we can see the PAFA, these genes, it work on the cellular protein metabolism and hormone stimulation. And the ASCC3, it work on the DNA duplex unwinding to make the DNA not tightly put together. It should be unwind and to do the transcription. And so the expression changed, the expression efficiency changed. Okay, the SNP story is still going on. If I have enough time, I will show what's going on recently. And another study we have is applied the commercial, the company named Agilent. They have a PIC gene expression microarray set is 44K means 44,000 genes being put on the microarray. And we tried it and to identify the mRNA expression profile. We collect the white blood cell. This time we put on our focus on the immune response. Because we know at this time, especially in the blastocyst, early blastocyst, the maternal and they are building the mother's uterus building the interface of mother and the embryo just during that time. So the embryo express some protein to inform its mother to prepare the uterus for them to implant. So we select 4 higher reproductive. I say that higher should be average, okay? Yeah, because the number born alive in average 9.3 plus minus 0.6 for 2 liters, and 4 lower reproductive dendrite cells is 5.1 plus minus 1.3 for 2 liters to compare 2 different stages. The first stage is 1 day after weaning, and the second stage is 7 days after conception. And at this time, is the time I mentioned, is the information from embryo to the mother. And for this time is just weaning, just wean. The pig is just wean, and the mother just from the crate, the farrowing crate, to the to waiting for next conception. So what we are going to try is if we can find the connection between these two, then maybe we can have more piglets when we check the protein expression during this time, because that's the time we make the decision on farm. These cells, I need to make it to take a rest for one cycle to gain more nutrition and some rest to have better performance for the next farrowing. And okay, so we the same way we do the microarray and using this one high and low to compare the high and low compare in this stage, also this stage. So we only focus on this and this because for this one is high in 1 day after weaning, and this one also 7 days after conception. So that's the connection, high to high. And this one is low, still low, the expression. So this for this part, we found totally the 2-fold change is 780 genes has the differential expression between high and low, high cell and high cell to get this 780 genes. And for low, this low reproductive cell expression here and here also, we can see the 1,158. And if we have a 4-fold change, it's in the parenthesis, it's 188, and this one is 69. For this group, the 700 or 69 genes, most of them concerning the metabolism and the genetic regulation. It's reasonable, because the nutrition and the management better for this, or they can absorb that kind of environmental change because their metabolism is better than the lower. And for this low reproductive dendrite cells, we found, and also here, this kind of genes, their functions, most of them concerning the immune function. At this time, they express more genes related to immune to response maybe to kill some embryo, not qualified embryo or something. Later on, I will talk about one protein is that kind of function. Or also, some of the genes concerning the inflammatory regulation, because probably they have inflammation in the mother. Okay, and there are so many information, so we need to make a database for them to collect all the information, put together, and more efficient later on, and also we can trace back everything back to the expression or the gene's function. So we have a database like this, the cDNA to protein and the gene annotation, the function annotation put together. So we can select on which one we are going to do the continual research. And also we have the pathway analysis to annotate which gene working on what kind of physiology process. Like an ATM gene, what does it do? With somebody put together to do some kind of physiology process. And the beta-2M is the one I mentioned. It concerning the natural killer cell. And evolve the natural killer cell to identify the non-qualified embryo and kill it. Yeah, that's the happened in the mother and the embryo interface. And we select 10 candidate protein markers, and then we make the monoclonal antibody production. And just one example, we use the beta-2M, this macro globulin as one example. We do the 2D protein expression like this for the total number bone 9.7 this cell number 2 and cell number 1 the same, and the cell number 3 and 4 the total number bone and the number bone alive is a lot less than this. And we found the beta-2M they have 8 spots expressed for the higher reproductive cell, and for the lower reproductive cell only 6, the 3 to 8. Or 3 to 8, the spots being expressed in the serum. So if in the serum, it's easier to check as an indicator. Then we design a 3-year experiment, actually 2. We need to know the cold season. We do the blind test, double blind. The lab don't know which cell, the serum from which cell. And the farms just do their own management, and they don't know which cell actually being take the sample. So they don't, both of them don't know just the experiment, doing the experiment, the collector knows which cell is which cell. So later on, we have the next parity liter size, the number bone alive of each the 10 cells being produced, next parity, and we see this and we get the answer out. So here the prediction of the beta-2M using the protein 2D gel to predict in the first day of weaning. The first day of weaning, when we do the prediction. And later, 120 days later, and we got the answer like this. And make it short. This part, we use the calculation, the blood calculation, to predict this cell next parity is high or low or intermediate. Yeah, actually, it depends on this kind of calculation, only 50% correct to the result. And if we do this prediction using beta-2M, it has 80% or 75%, depends on what kind of indication you have. yeah, about 75 to 80% correct. We do this co-season test 2 years, 2 times. Yeah. And we found that it's kind of, we can maintain 70 to 80% accuracy. But the hot season is challenging. For the calculation, we only have 40% correct. And for the prediction using beta to M, drop down to 55% correct. So it means there should be another probably several markers we need to develop and do the same thing and put them together to find out when the first day of wean, which cell should get into the next conception, 5 or 6 days later, or some of the cells should take a rest for 1 cycle and come to the next conception. For the conclusion, we have the whole things from 10 years ago with the functional genome microarray study, SNP prediction discovery, and the SNP validation, then go to the breeding animals to select the genetically progress to do the like a candidate gene, but we have more SNP markers to use. And for this part, we do the protein annotation, monoclonal antibody production, then do the cell management check or cell selection on farm after weaning. So, do I have time for that? No more? Okay, yeah, that's it. Yeah, I just stop it here.
Well, thank you very much. Are there any, this was time for one informative question. Yeah.
More information about the use language more used in Japna.
In Taiwan? Yes. Yes. We use the nitrates as the female line, the crows, Yorkshire boar, to have the 2 breed crows for the female. But after 1997, the outbreak of FMD, we stopped the export, the pork export to Japan. So to the domestic market, we have feed the nitrates major as the female, just 1 breed, crows the Duroc directly.
So Duroc you terminate
yes, terminal sire is Duroc, major.
Well, thank you very much. Just show our appreciation.